Hi, this is Matthew Kenslow, and for my Microbiology Lab's research project, I analyzed bactericidal versus bacteriostatic properties when different types of concentrations of rubbing alcohols are independently applied to gram-positive and gram-negative bacteria. Introduction. Isopropyl alcohol and ethyl alcohol are two main types of alcohol found as the active ingredients in different types of rubbing alcohols. Isopropanol simply has an extra carbon and two extra hydrogens in its chain and is therefore longer, plus the hydroxyl is on the 2-carbon. Rubbing alcohol can be used as first-aid antiseptics and cleaning agents. Isopropanol is optimal between 60 and 90% concentration and is more efficient against bacteria, whereas ethanol is efficient against viruses. Previous studies have concluded bactericidal activities with ethanol. Ethanol had killed most bacterial pathogens within 15 seconds. In this study, ethanol and two different concentrations of isopropanol are used to test their effectiveness against gram-positive and gram-negative bacteria. Staphylococcus epidermidis and Escherichia coli are used as the gram-positive and gram-negative respectively. The main difference between gram-positive and gram-negative is that gram-positive bacteria has a thicker peptidoglycan cell wall, but gram-negative bacteria have an outer plasma membrane in addition to the one underneath. Previous studies had also shown that E. coli had a higher reduction factor relative to S. epidermidis. Other studies show that gram-positive bacteria have more resistance. It had also been deduced that either alcohol-based solution will be readily bactericidal versus bacteriostatic when it comes to vegetative forms of bacteria, among other antimicrobial effects, provided that the concentration stays above 50% by volume. Alcohol in general should work in the disinfection and antisepsis of microbes, not just bacteria, due to it exhibiting quick, broad-spectrum activity. Additionally, alcohol works against E. coli by denaturing its dehydrogenases, which are enzymes of E. coli, but should not be used as sterilizing agents since they lack sporicidal activity. This experiment is performed to answer the following questions. Does rubbing alcohol act more as a bactericidal agent or a bacteriostatic agent? Can ethanol be efficacious against either or both categories of bacteria? Does the concentration of alcohol make a difference in effectiveness? By the same principle, what can be said about the quantity of alcohol added? Does having a bulky polar molecule add or take away from the effectiveness than the ambipathic linear one, i.e. isopropanol or ethanol respectively? Materials and methods. You would need 9 nutrient broth tubes with a culture tube rack, reactivated Staphylococcus epidermidis, which had previously been lyophilized. This will serve as the gram-positive bacteria. Reactivated Escherichia coli, which had previously been lyophilized. This will serve as the gram-negative bacteria. Sterile transfer pipettes, a source of a flame such as a paraffin candle, including a match or a lighter. Grease pencil, personal protective equipment such as gloves, apron, and a 10-part bleach disinfectant with paper towels. Here's the protocol that I came up with. 1. Take and label 9 nutrient broth tubes number 1 through 9 and date. Into tube 1, pipette a half milliliter of E. coli. Into tube 2, pipette a half milliliter of S. epidermidis. Into tube 3, pipette in half a milliliter of E. coli and 10 drops of 70% isopropanol. Into tube 4, pipette a half a milliliter of E. coli and 20 drops of 70% isopropanol. Into tube 5, pipette a half milliliter of E. coli and 10 drops of 91% isopropanol. Into tube 6, pipette in a half milliliter of E. coli and 20 drops of 91% isopropanol. Into tube 7, pipette in a half milliliter of E. coli and 10 drops of 70% ethanol. Into tube 8, pipette in a half milliliter of E. coli and 20 drops of 70% ethanol. And into tube 9, Pipette in a half a milliliter of S. epidermidis and 20 drops of 91% isopropanol. Incubate for 24 hours, examine, and take pictures. Disinfect the area. Wait an additional 24 hours, examine, and take pictures. Wait an additional 24 hours, examine, and take pictures. Here are my results. Here are the pictures that I've taken after approximately 24, 48, and 72 hours. Here's what was exhibited in each tube, and now I'm going to decipher them right here in the discussion section. As expected, due to turbidity, there was growth in both controls, with the greater amount of growth being in the one with the gram-negative bacteria, E. coli. When using 70% isopropanol, there was little growth relative to the E. coli control. When using 91% isopropanol in the gram-negative bacteria tubes, they both were much clearer, with the clearest of the two being the one with a greater quantity of isopropanol, that is 20 drops instead of 10. This appears to validate the expectation that the greater concentration of alcohol means a less amount of growth, if any. When using 70% ethanol, however, there was growth in both tubes, but oddly enough, both growths were floating in the upper half. This may be because of ethanol having a longer carbon chain away from the hydroxyl group. So you see here's the carbon chain, and the hydroxyl group, or the polar part of ethanol, is way over here. Isopropanol's hydroxyl group is right here in the middle. Basic organic chemistry tells us that hydrocarbon chains are hydrophobic. They hate water. In the tubes, it was still soluble nonetheless, as it is a small amphipathic molecule, and it did not form an immiscible layer above the broth. The fact that there was nearly as much growth as the controls substantiates the background research when the article said that ethanol is more efficacious for killing viruses than bacteria. Finally, when using 91% isopropanol for the gram-positive bacteria, it did not appear to have any growth, which, according to my experiment, seemed to have counterclaimed the article that said gram-positive bacteria are more resistant to bactericidal effects. Going back to my initial questions, does rubbing alcohol act more as a bactericidal agent or bacteriostatic agent? To an extent, viewing the results of my experiment, neither of such agents. The two best antibacterial results were the two using the most drops of the highest concentration of isopropanol. The two worst results were the two using 70% ethanol. Can ethanol be efficacious against either or both categories of bacteria? My experiment concludes no. Does the concentration of alcohol make a difference in effectiveness? My experiment concludes yes. A 91% concentration had a greater effect than 70%. By the same principle, what can be said about the quantity of alcohol added? My experiment concludes 20 drops were more effective in both parts of my experiment using isopropanol against E. coli versus 10 drops. And finally, does having a bulky polar molecule add or take away from the effectiveness than an amphipathic linear one? My experiment concludes that it added to the effectiveness. The bulky polar molecule utilized was isopropanol. The linear amphipathic one was ethanol, and my experiment shown that it was the least effective. Since I was limited on resources and time, I was unable to do additional experiments in response to my results. For future experimentation, repeat the protocol for tubes 6 and 9. After incubating for 72 hours, take both of them and inoculate them separately in a tube of nutrient broth. Do not treat it with any additional 91% isopropanol. After an additional 48 hours, if there is still no growth, then that means the agent, including its concentration and quantity, works best against bacteria. If there is regrowth of the bacteria, then that would mean the agent, including its concentration and quantity, was just bacteriostatic the entire time. This may be saying that, whenever applying the 91% isopropanol anywhere, the bacteria will eventually regrow once the effectiveness wears off. In the interim, isopropanol is a great disinfectant, but not a sterilant. Here are my references, and I really appreciate you taking the time to listen to this. Have a nice day.